Hello, this is Dr. Steve Feldman, Professor of Dermatology at the Wake Forest Baptist Medical Center in Winston-Salem, North Carolina. I'll be discussing the publication, The Totality of the Evidence Approach to the Development and Assessment of GP 2015, a proposed etanercept biosimilar by Dr. Strand and colleagues. This study report was published in the Current Medical Research and Opinion. I selected this article to discuss the totality of the evidence concept. Physicians may be unfamiliar with the totality of the evidence concept. The goal of this review is to describe the inherent variability that is natural to biologics, and using the proposed etanercept, a tumor necrosis alpha inhibitor biosimilar GP2015 as an example, provide details on the type and extent of analytical, preclinical, and clinical data considered by regulatory authorities with respect to the approval of biosimilars. Physicians have been trained to look at clinical data primarily when making choices about which drug to use. For biosimilars, however, physicians need to be aware of the complete data package, the analytics, preclinical, and clinical data that's considered by regulatory agencies when concluding whether a proposed biosimilar is approvable or not as a biosimilar. The totality of the evidence includes all analyses and performed trials used to approve the product and justify use of the biosimilar in all indications for which the reference medicine is approved. This article is an overview of the stages of the in-depth process of developing a biosimilar from its reference medicine, in this case, the etanercept. Biosimilar development involves a number of stages. The initial stage examines multiple batches of the reference medicine, and they're characterized to understand structural and functional attributes and determine the variability of post-translational modifications over time and from batch to batch. Data then set the development target and boundaries of acceptable variability of the biosimilar. Stage 2 involves target-directed development of a manufacturing process for the biosimilar molecule. The final stage includes confirmation of high similarity, comparing structural and functional level to determine whether the biosimilar molecule is essentially the same as the reference molecule. Then preclinical properties, toxicology, tolerability, and non-human pharmacology are compared using relevant in vitro and in vivo models. Final phase 3 confirmatory clinical efficacy and safety trial is conducted in a relevant population to allow detection of clinically meaningful differences between the biosimilar and reference medicine, should such differences exist. These analytical data serve as a foundation of the overall comparability exercise and the totality of the evidence concept. Discussion of confirmatory study is to use a clinical indication, primary endpoint, study duration that is the best chance of detecting differences in efficacy or safety between the biosimilar and reference product if such differences exist. Biosimilars also offer an opportunity to learn more about biologic medicines in general. For example, how changes in the critical quality attributes of a biologic correlate with clinically relevant outcomes. Let me share my thoughts about this study and an analysis of it. This study highlights the tremendous extent to which biosimilars are studied to assure that they will function the same way as the innovator biologic. Biologics are very complicated molecules. No one can create perfect copies, but we can make sure they're highly similar, so similar that we would not expect the biosimilar to perform any differently than the innovator product. I expect the detailed description this manuscript offers of the studies done on biosimilars will help assure doctors and patients that they can expect the same extraordinary, life-changing benefits from biosimilars that we have seen from the innovator biologics that the biosimilars are trying to copy. I expect we will see, pending any legal challenges, rapid uptake in the use of biosimilars driven by payers and largely accepted by the medical and patient communities. Because biologic medications are too difficult to perfectly copy, there will always be some degree of uncertainty, even with the innovator products. That degree of uncertainty has not bothered us since the introduction of innovator products. It may be that the greatest uncertainties that remain are largely practical legal issues of bringing the biosimilars to market.